I am Maria-Elena Torres-Padilla and we work in the IGBMC in Strasbourg, France.
Hello, I'm Adam Burton. I'm a postdoctoral researcher in Maria-Elena's lab.
Today we will present to you the work that will be published in this issue of Cell Reports and that we did in collaboration with Ernesto Guccione from the IMCB in Singapore. Our lab is mainly interested in understanding how the pluripotent cells form in the embryo as the embryo develops, and in particular how this is regulated through chromatin. This is very important because the embryo is undergoing epigenetic reprogramming.
In order to gain a greater insight into the role of chromatin-mediated processes during preimplantation development, we decided to perform a single-cell gene expression analysis of chromatin-modifying enzymes.
To do this, we collected embryos at each stage of development from oocyte to blastocyst and separated them into single cells. The single-cell cDNA samples were then analysed for expression patterns of a cohort of chromatin-modifying enzymes using fluidigm biomark technology. This microfluidic space chip allows the simultaneous and quantitative analysis of 48 by 48 or 96 by 96 genes per cell to gain unprecedented insight into the regulatory networks of chromatin-modifying complexes during preimplantation development. We found that single cells cluster well into their particular stage of development from the oocyte to 8-cell stage. This is also apparent when mathematical modelling of our dataset is performed, generating an epigenetic landscape of preimplantation development. The early stages form distinct energy wells, indicating more stable states, while later stages of development are not so well defined. Thus, we conclude that during preimplantation development there is a dynamically evolving chromatin architecture, particularly at the early stages.
Using the power of our single-cell expression analysis, we were able to identify genes with a heterogeneous expression pattern at a given stage of development.
We found that one gene in particular, PRDM14, had a highly heterogeneous expression pattern at the 4-cell stage of development. This was also conserved at the protein level, with punctate nuclear expression in 2 of the 4 cells of each embryo. We hypothesized that based on its heterogeneous expression pattern at the 4-cell stage, as well as higher expression in the inner cell mass lineage, PRDM14 could play a role in lineage allocation during preimplantation development. To address this, we performed the following experiment. We overexpressed PRDM14 by injection of its mRNA in one cell at the late 2-cell stage, along with GFP as a lineage tracer. We then followed injected embryos to the blastocyst and determined whether GFP positive and thus PRDM14 injected cell progeny were enriched in the inner cell mass. Indeed, we found a significant bias for PRDM14 injected cells to form the inner cell mass lineage as opposed to the outer trophectoderm, suggesting a role for PRDM14 in cell fate determination and generation of the pluripotent inner cell mass.
So, in conclusion, our data have identified novel regulators of pluripotency in vivo. On a more global level, they stress the importance of quantitative analysis of gene expression in single cells, and they have shed light to the epigenetic networks governing preimplantation development.
Thank you, and we hope you found our paper of interest.